thank you to all the organizers for inviting me today. I'm very excited about what I see here. Congratulations to what you've built. Congratulations to the opportunity you have to go forward. I think that we are positioned, and for those of you that have to leave early, I can give the entire talk right now. The answer is, I think we're positioned at the beginning of an enterprise in which the KI and Sweden could lead, frankly, to make medicines a new way that will help more people than we can count in the next few decades. So that's the future. I'll start my talk by going a little bit backwards in time. So this talk is a personal one for me. As you heard, I'm a recovering neurosurgeon. I don't do it anymore. But I've spent 30 years studying inflammation and immunology. And although it would be nice if I could say truthfully that Grandma came to me in a dream and said, you should combine these as a scientist, that's not what happened. It happened by a series of accidents. And what happened from those accidents, as Jan just explained, was a concept that if we understood how individual neural circuits could control individual aspects of immune responsiveness, then it should be, and we knew the mechanisms, then it should be possible to design devices to control the nerve, to control the mechanism. And that's what I call bioelectronic medicine. The key is it's a process. Inventing a process or developing a process is more important than developing a product. The process will lead to infinite new products and infinite new discoveries. Bioelectronic medicine is a process. The ability to make it work goes back to understanding fundamental mechanisms. And one mechanism we have to begin to understand first is, what is a reflex? So the father of all reflexes was René Descartes. And history has it in a story that's probably apocryphal, that he was walking through the king's gardens in Saint-Germain, and one of the statues lunged at him. And the statue was built to lunge at him. The king thought this was a riot, that he could have a hydraulic system from the sidewalk to the statue, and when the guests would step on the sidewalk, a hydraulic system would make the statue move at the unsuspecting guest. And so the king would watch his guests become terrified, and he thought this was great. René Descartes was fascinated by the idea of a hydraulic principle controlling behavior. So what René Descartes thought was maybe what nerves do is take a change in the environment, heat in this case, a fire, or pressure on a stone, or pressure on your arm, and that nerves respond to the environment with a reflex response that causes a behavioral that you can measure. Now, the concept of nerves was well known, and there was no way for René Descartes to know how they worked, and he thought hydraulics was as good an explanation as any. That changed, of course, when Ramon y Cajal discovered and showed these beautiful pictures of neurons. Now, everyone knows that Ramon y Cajal was able to visualize these neurons with Golgi stains, but what some people have forgotten is that another major discovery of this work was that the nerves could only send signals in one direction. So that sensory nerves send signals into the nervous system, and motor or efferent nerves send signals out. And when you understand that, now you can go back to the lunging statue or the principle of reflexes and understand that nerves send signals in a single direction, and that when a signal goes in and activates a signal coming out, that's a reflex. Now, Sir Charles Scott Sherrington dedicated his entire life to understanding reflexes. And he understood something else that we tend to overlook, I think, sometimes in the modern era. Almost every cell in the body is innervated. Almost every cell in the body is encased in a neural net, either a sensory neural net or a sensory and motor neural net. And it's complicated because sensory neurons in this net can send signals actually in both directions, sensory and motor. And so here, this is a picture of one of Sherrington's photomicrographs of the masticatory organ of a jellyfish. And he understood that that this complex chewing response or food intake response in the mouth of the jellyfish was regulated at the single cell level by individual neurons. And Charles Sherrington proposed that you should think of a reflex as having an afferent or sensory arc, a processing center, and a motor arc, and that the motor arc would transmit this information back to the area that the sensory change originated. And he said, if you have to understand this simple reflex as these three components, but you also have to remember that there's no such thing, because all the reflexes are connected, like that complicated diagram I showed you of the jellyfish mouth. Nonetheless, by asking mechanistic questions about simple reflexes of three arcs, you can do very complex physiology. And in fact, in this same era, Harvey Cushing, the father of neurosurgery and endocrinology, not infectious diseases, sorry, Jan, Harvey Cushing actually studied the role of the vagus nerve in regulating complex cardiovascular physiology. To do this, he put a balloon in the brain of a dog and raised the pressure in the balloon by raising the fluid sac here. And what he showed is what's now known as the Cushing reflex. As intracranial pressure increased, blood pressure would increase, heart rate would slow, and eventually respirations would stop. And what he did in these animals, which I didn't realize until recently, is he also did vagotomies. He studied the role of the vagus nerve. He cut the vagus nerve and showed that the effects on the heart and blood pressure changed. So this is ancient history, right? But as you're going to see, it's still very current. Again, a contemporary of Cushing's, Paul Ehrlich, became very, very sick and with tuberculosis. And while he was recovering in bed, he conceived of magic bullets. He was a dye chemist, and he would screen chemical libraries to pick a perfect dye for a cloth. And he wondered if he could screen for molecules that would hit specific receptors. He invented the word receptor and chemoreceptor. He invented the drug screen of chemical library screening for drugs, and he invented chemotherapy with the first successful drug originally named Ehrlich 606, but later salvarsan, which was the first specific synthetic treatment for a specific disease, syphilis. Basically, what we need to remember from that point to today is that the pharmaceutical industry has not changed. From the time of Ehrlich to today, the pharmaceutical industry is focused on discovering, developing, producing, and marketing drugs as therapy. Currently, it's $1 trillion in annual sales worldwide for the prescription market, and the strategy is the exact same one that Ehrlich proposed. Identify a molecular target, screen the candidate molecules to minimize but never eliminate toxicity, and then sell them. That is the pharmaceutical industry today. That's the strategy. The tactics we today call molecular medicine. The libraries have become more complicated. We screen for monoclonal antibodies, we screen for genes, we screen for complex chemical libraries, but it's still the same process. Identify a molecular target, screen for drugs, and minimize toxicity. As far as the vagus nerve, by the end of the 20th century, we knew a lot more too. We knew that it was comprised of not a single copper wire, as some things that are even being written today would lead you to believe. It's more like 100,000 copper wires. It's 100,000 individual fibers in a human. About 80% are sensory and 20% are motor. And it touches all the organs of the body that are important to life and the ones you never think about. Because it touches these organs and because it provides a reflex basis of controlling organ function, the physiologists of the 20th century worked out in great detail the vagus nerve's role in controlling virtually all the organs: heart rate, blood pressure, respiratory rate, insulin secretion, GI function. And the list goes on and on and on. But what was not on the list at the end of the 20th century was the immune system. The immune system was thought to be a non-innervated organ. The immune system was believed to be a circulating organ. And although that the immune system cells spend some time in tissues that are innervated, like the spleen and lymph nodes and others, it was not believed that the neural signals to those organs would control the function of those cells, at least not in any specific way. So that's how things stood at the end of the 20th century. At the time when I was training to be a neurosurgeon, and I met the patient Jan referred to, and her name was Janice, and she had crawled across the kitchen floor when her grandmother was cooking spaghetti. And when grandmother turned to drain the boiling water in the sink, she tripped over her granddaughter, and she spilled the boiling water on her 11-month-old baby, granddaughter. So I met Janice with this horrible scald burn over 90% of her body and cared for her for a month. And she, in fact, was septic, and she did go in and out of septic shock, but she was scheduled to go home. We were beginning discharge planning because she'd done incredibly well after skin grafts and a month of the ICU. And for inexplicable reasons, one day while she was having lunch in her nurse's arms, rocking in her nurse's arms, her eyes rolled back in her head and she died. And I was standing in the doorway. I ran in, I put her in my arm, I gave her mouth to mouth, called a code. There was a cardiothoracic team in the hallway. By coincidence, they were there in seconds, and everything went flawlessly except that Janice was gone. And so this case haunted me because it couldn't be easily explained by infection because we never found any infection, and it couldn't be explained by endotoxin, which was the dogma at the time for acute shock and death, because we didn't find any endotoxin or gram-negative bacteria. So I did go in the lab with the groups at Cornell and Rockefeller, Steve Lowry, Tom Shires, Tony Cerami, Bruce Beutler, and others, and began working on the idea that a molecule called TNF could have, in fact, been the molecule that killed Janice. And what we found was that TNF was sufficient to cause shock, and that in this experiment I'm showing here, that monoclonal antibodies to block TNF could prevent inflammatory damage in baboons. The baboons in these models were infected with E. coli. The animals made large amounts of TNF and died. Animals, however, that were treated with monoclonal anti-TNF were saved. This was the first published paper using monoclonal anti-TNF as a therapy, and the data are shown here. So here's an animal that died within hours after receiving E. coli. These other two animals received monoclonal anti-TNF and were protected. So all of these animals have replicating bacteria in their bloodstream. This gives direct evidence that the TNF was the problem, not the bacteria. So this paper's been cited thousands of times, and it's been extraordinary to me to see these antibodies now used in millions of patients. And it's extraordinary to think on the business side of it that these antibodies and the family of antibodies of biologics to block inflammation against other inflammatory cytokines represents more than 5% of the world's pharmaceutical industry and growing. It's a huge opportunity to treat inflammatory diseases. On the mechanistic side, we came to understand that cytokines are both necessary and sufficient to cause some diseases in humans. And I call this the cytokine theory of disease. It doesn't say that all cytokines are bad. The germ theory doesn't say that all germs are bad. Just that in some situations, a specific cytokine in a specific patient can be necessary and sufficient for the disease. And today, anti-TNF and anti-IL-1 are approved drugs in humans. Clinical trials will hopefully be started soon on anti-HMGB1, a cytokine that we worked on extensively here with Ulf and Jan and others at the KI. These molecules hold promise for not only helping patients, but also for giving an insight that the balance between how much cytokines are produced tips the balance between health and disease. So this is a point I'll come back to in a minute, but if you're an engineer, this would be the critical controller point in a closed-loop system. This step here between how much cytokines are being produced would be a gate or a regulator tipping the balance towards health or disease. And this is where evolution has a tremendous advantage in selecting nerves to control this step, and I'll come back to that in a few minutes. But in the meantime, in the early 1990s, I moved to the Feinstein Institute, and I wanted to develop a molecule that would work at this step to block cytokines that wasn't an antibody. Monoclonal antibodies are great, but they're not perfect. They only work in about half the patients, typically. They're very expensive. They require injections. Some of them have black box warnings, which means you can die from it. So there are other alternatives still needed, and I wanted to try my turn at making molecules to do this. And so as a result of a screen, the 14th molecule in 1992 was this one, and we didn't want it to be named 1492 because Columbus had that already. So we changed it to 1493. we took the liberty of adding a year. And this molecule turned out to be extremely good at blocking cytokines. It turned off the production of TNF and IL-1. It prevented disease signs and symptoms in animal models. And this was a time where I want to make a point that I love doing science and I love having mechanistic understandings. But to me, the most gratifying thing is when you move your science into a patient so a patient can benefit. And that requires a few more steps that I'll talk about on and off in this talk. One is that in order for an idea or a discovery or an invention to help a patient, it's going to require lots of money and time and work. And that requires having some knowledge of intellectual property and business development, or having people that the scientists work with having that knowledge. It's a critical gating step in succeeding or failing. And I tell the Feinstein board members every quarter that understanding the for-profit and business side of science to help patients is on the critical path of our not-for-profit mission to produce knowledge to cure disease. It's a critical point. So we developed a patent estate around CNI 1493, and with that patent we were able to start a company, the first company I started called Cytokine Networks Incorporated. Cytokine Networks later did, with that molecule, a phase 1 clinical trial of CNI 1493 at Harvard. Cytokine Pharma Sciences, they changed their name, Cytokine Networks to Cytokine Pharma Sciences. They sponsored this trial at Harvard. These are patients with cancer who are getting IL-2 infusions, and their TNF levels are rising because the IL-2 turns on the cytokine cascade. But when we treated these patients with CNI 1493, the cytokines were blocked. So here was a molecule we'd invented that could block TNF in humans, and we were really excited. And this molecule went on for many, many years. Eventually, it was dropped because it had side effects on local inflammation if it leaked out of the IV or the injection. So that molecule wasn't developed further, but we thought we knew everything about it. It was a p38 inhibitor. It blocked the uptake of arginine in macrophages. But we didn't know everything about it. And this was, so if Janus was one event that set this up, a second was sort of an accidental finding in the lab. We were studying animals with stroke and injected CNI 1493 in the brain of animals to turn off the TNF in the brain. What we didn't expect is that it would turn off the TNF in the lungs, the spleen, the gut, the other organs of the body. This made no sense whatsoever. I can play that again if it was not too dark. So when you put the drug in the brain in teeny amounts, it turned the TNF off. But when we looked in the other organs, it was also turned off in the other organs. And this really made no sense, because we'd only put a vanishingly small amount of CNI 1493 into the brain. This experiment actually has some relationship to KI as well, because we were interested in the effects of this drug in the brain, having collaborated with Jan Anderson on studying meningitis. And we were looking at the effects of CNI 1493 in the brain on blocking cytokines in Jan's meningitis model, and then moved on to do a stroke model. So it was in the course of those stroke models that we found this amazing discovery. How could a few picograms of drug in the brain turn off the TNF in the spleen and the lungs? We repeated the experiment many, many times, published it in the JEM. Tom Bernick did this work. You can see here the IC50 for intravenous or systemic 1493 is on the order of 0.2 milligrams per kilogram. The IC50 or the EC50, when you put the molecule in the brain, was 30,000-fold more potent. So there's something about the molecule in the brain that turned on signals to the body's organs to turn off TNF. Months and months of thinking about this proved very frustrating, frankly. And finally, remembering Harvey Cushing, we cut the vagus nerve to see if could these signals be traveling in the vagus nerve. And when we repeated that experiment, in fact, now the signals going from the brain to the organ were blocked. And so the vagus nerve was carrying the signals about the off switch, the presence of the drug in the brain, to the body's organs. But that's all we knew. We had no idea how that could possibly work. So basically, I'm in my neurosurgery clinic at that time. I was still operating on patients, tapping on the knees of various immunologists if they came in with their herniated discs. And I will tell you, this cartoon is broken, but if it was running, you would see that when the neurosurgeon hits the knee, the immunologist kicks the neurosurgeon in the head. And maybe it was something like that benefited me, because we pored over the literature and could find nothing about the immune system and the vagus nerve until we found this paper by Linda Watkins, which was virtually never cited. And what Linda had showed was if she injected IL-1 into rats or mice, they got a fever, which was, of course, a known effect. What was not known, and what she discovered, is if she cut the vagus nerve, the fever was gone. This meant that the vagus nerve was telling the brain to start a fever because it saw the IL-1 in the body. So thinking about this, again, for weeks and months, I wondered if we'd in fact discovered a reflex, and that Linda had proposed an afferent arc of a reflex, because we knew a lot about TNF and IL-1 causing fever and anorexia, and I wondered how many of those effects might actually go through the vagus nerve. And perhaps by putting 1493 into the brain, we had discovered the motor arc that would go back and counter-regulate the inflammatory cytokines at their origin. This at once presented an obvious experiment to me that could answer the question, and that would be to simply not put 1493 in the brain, but just add an electric signal to the vagus nerve directly using an electrode to send this off switch back into the body. And so that's what we did. And Ludmila Borovikova, a postdoc in the lab, did these experiments and showed that if you give endotoxin to a mouse or rat, you get a lot of TNF. If you electrically stimulate the vagus nerve, the TNF is blocked. If you cut the vagus nerve, you lose the brakes and you overproduce TNF. So there's a tonic inhibitory effect at rest. Here, if you look at blood pressure, same thing. Animals go into shock from making too much TNF. Cut the vagus nerve, they go into shock more quickly, and they're protected against shock and TNF by electrically stimulating the vagus nerve. So I described this concept of an inflammatory reflex, reasoning that the immune system has a complex regulatory network of molecules that float in the bloodstream to turn off TNF, like IL-10, spermine, glucocorticoids, hormones, MSH, and other factors. And that maybe what we had discovered was the neural component, the neural reflex basis of counter-regulating cytokines. And this has several immediate advantages, right? Because nerves can go to a discrete place. You can do something in the liver and not the heart. And nerves have different neurotransmitter mechanisms, and nerve mechanisms are short-lived. If you give a dose of steroids to a patient, it's going to circulate for hours. You could electrically stimulate a patient and have an effect for just a few minutes. And these all seem like important advantages that we described in this paper called The Inflammatory Reflex, which was basically a hypothesis paper backed by a little bit of data at that time. But it was a strong basis for thinking about the question of whether electrons might be able to replace drugs by blocking TNF and other cytokines. We have a good understanding of how reflexes work in neuroscience and that they're specific. Now we have a good understanding that cytokines are a reasonable target through the cytokine theory of disease. And now we have evidence that neural circuits can control cytokines. So in the late 1990s, I sketched on the back of a napkin the idea of embedding patients with a device on their vagus nerve. Patients with rheumatoid arthritis and Crohn's disease who were being treated with monoclonal anti-TNF as a way to turn off the TNF or IL-1 in their body as a therapy. We wrote patents on this idea, which were issued. And frankly, there was already at that time patients receiving vagus nerve implants for other diseases like epilepsy, and I was ready to do the clinical trial in 2002 or so. But that wasn't possible really for a couple reasons, and the most important one is we didn't really understand all the mechanisms. And so from then till now, we've mapped the mechanisms of the inflammatory reflex in order to do the clinical trials. And I'll summarize 500,000 man-hours of work over 20 years in just a couple minutes. But basically what we've learned is that signals arising in the body can ascend in the vagus nerve to the brainstem and activate in a black box, an unknown neural circuit in the brainstem, probably through the nucleus tractus solitarius to the dorsal motor nucleus of 10, to send signals back down the vagus nerve that can culminate on the spleen. And the spleen is important for these understandings because the spleen is the major source of TNF during endotoxin animals in mice, endotoxin infusions in mice and rats. So we wanted to map a specific circuit, and the specificity was further enhanced because the spleen has one artery in, one vein out, and one nerve in. So we could map all the mechanisms and connect the dots. And when you do that, what you do is you can follow the vagus nerve signals down into the spleen, and the signals end on T cells. Now this T cell is green because it is a transgene with GFP linked to the expression of choline acetyltransferase. So this is a T cell that can make acetylcholine. Now this work was done by co-first author Petar Olofsson and Mauricio Rosas-Bellina, and it turned out to be an extremely important paper because these T cells are unique, and this paper represents the discovery of these T cells. These T cells represent about 1% of the T cell population in the spleen, probably similar numbers in the lymph nodes and blood. Peter went on to characterize these T cells, and you can't read this, but the green spike here is the expression of CHAT. So this is choline acetyltransferase, the enzyme that makes acetylcholine. So the vagus nerve signal travels down the vagus nerve to the spleen, to the splenic nerve. The splenic nerve is adrenergic. The adrenergic signal there turns on these T cells to make acetylcholine. These cells are extremely important, and this is not just me saying it. The rest of the world seems to think so too. I checked yesterday, and this paper that Peter wrote is in the 99th percentile of 261,041 articles of a similar age in all journals, and it's in the 97th percentile, ranked second for Nature Biotechnology. So these are very important cells as part of the inflammatory reflex and in understanding the counter-regulation of inflammation, and they're very important cells in the regulation of blood pressure, but we'll talk about that another time. Once the acetylcholine is made, it signals through a specific receptor called alpha-7. This is evidence of that from a paper done by a postdoc on the lab, Hong Wang. If you give endotoxin to wild-type animals, they make TNF. Vagus nerve stimulation blocks it. The Shan animals, the alpha-7 knockout animals, you see two phenotypes. The first is that when you give endotoxin, they make more TNF. Again, they've lost the brakes on the TNF control. And second, when you give vagus nerve stimulation, there's no inhibition. So this single receptor, which is expressed in human cytokine-producing cells and can be targeted in whole blood human monocytes, is incredibly important in regulating cytokines. Ben Liu elucidated the mechanism of alpha-7 signaling by how does it block the cytokines, and it turns out it's primarily an effect of alpha-7 expression on mitochondria, not the cell surface. So an activated macrophage upregulates nonspecific cation transporters that bring the acetylcholine into the mitochondria, and alpha-7 is mostly expressed on the mitochondria, where it downregulates the release of mitochondrial DNA, prevents the activation of the inflammasome, and hence you see two separate mechanisms for blocking cytokines. This one prevents inflammasome-dependent cytokines like IL-1, IL-18, and HMGB1, and a second mechanism targeting NF-kappa B prevents the transcription of the NF-kappa B-dependent cytokines like TNF. So that's 20 years. In one little movie, the signals go down to the splenic nerve from the vagus nerve. Inside the spleen, norepinephrine is released. The norepinephrine activates those T cells. The T cells, they can make acetylcholine. They make acetylcholine, which in turn interacts with alpha-7 on the cytokine-producing monocytes and macrophages and shuts off inflammation. Now, that's probably how it works in rheumatoid arthritis, because you have cells passing through the spleen, 20% of cardiac output all day long. And as the cells pass through the spleen and get these signals from the vagus nerve-dependent signals, the cells come in to the spleen. Think of them as M1 or inflammatory monocytes. They get the signal in the spleen and leave as M2, tissue-protective monocytes. So now when these cells get to the inflamed joint or the inflamed bowel, they're M2 tissue-protective, they don't cause damage. And that's, in fact, what we're seeing in patients. And so, although I wanted to do that, as I said, in 2002, 2003, the time finally did come to try these experiments using vagus nerve stimulation in rheumatoid arthritis and Crohn's disease patients actually quite recently. Now, the key, as I said before, is evolution would have had hundreds of millions of years to acquire the information to have a precise feedback control on this critical step. And so, in fact, there's very good evidence that single neurons in the C. elegans, in the roundworm, that single neurons can control the inflammatory potential of gut macrophages in the roundworm. So we humans have retained that ability, acquired through hundreds of millions of years of evolution, to have feedback control on specific immune responses through neurons. We took this as an opportunity to put vagus nerve stimulators in patients and turn them on. So that was a second company that we licensed. The patent I showed you was licensed to a company first called Innovative Metabolics, then called SetPoint. SetPoint sponsored the clinical trials led by Paul Peter Tak and Ralph Zitnik. And what you're seeing here is the first question we wanted to answer in the clinic is placebo effect, because everybody wants to talk about placebo effect. So we said, let's do that first and put it out of the way. These are patients who were having surgery to have a vagus nerve implant for epilepsy. And so they'd never had a device before. And so before anesthesia was given, we drew their blood, and this was work done in the lab primarily by Sangeeta Shivan. And Sangeeta measured the whole blood cytokine response. And these is before anesthesia and after anesthesia, but before the vagus nerve stimulator was turned on, and then turn on the vagus nerve stimulator, and you see the TNF goes down. So this is the first evidence in humans that targeting a specific nerve can control a specific component of the immune response. We then took this knowledge and applied it in a clinical trial of patients with severe rheumatoid arthritis. And it's a much longer story told by others who are full-time rheumatologists, but these are very sick patients because that was the criteria that was decided upon for the trial design. In rheumatoid arthritis, there's an inflammatory pannus that destroys the bone and cartilage, and the thinking is that the production of cytokines can make the damage in this pannus worse, and the blocking cytokines is beneficial. So patients were enrolled in the trial with severe rheumatoid arthritis, and they received an implant implanted under their collarbone with a lead up to the vagus nerve, and this thing was reprogrammed. So it was a device from Cyberonics that out of the box fires 5 minutes on, 5 minutes off all day long. But after the 20 years of work in animals, we knew we didn't have to do 5 minutes on and 5 minutes off all day long. We knew that would actually probably be a bad thing. We realized that you could fire, and this is critical work that Peter did when he was in New York. We realized that you could fire the stimulator for 1 or 2 minutes, and that the macrophages that got turned off would stay turned off for 24 to 48 hours. So SetPoint reprogrammed this device to fire it for a few minutes, a few times a day, and put that in patients, and the responses were absolutely phenomenal. Within weeks, in the first patients, there was a big clinical effect. And so I flew to meet the first patient. So that is me on the left, looking younger, obviously. The second guy in, the tall guy, is the patient, and he had been home on his couch for 10 years, unable to work, unable to play with his kids, and he enrolled in the trial. This is his doctor. This is her boss. He was the head of medicine for pretty much all of Bosnia, and this was the electrophysiology technician in the hospital. I asked the patient, "Do you feel better?" He said, "Yes." And I said, "Do you have more energy? Did you gain or lose weight?" He said, "No, everything's fine. I just don't have pain anymore." And his story was that he, during the trial, was feeling so much better that he started playing ping pong. The ping pong went so well that he went outside and started playing tennis. Now, he was completely deconditioned. He hadn't been off the couch in years. He promptly injured his leg, which was a disaster because it counts as a painful joint and damaged his score. So his doctors told him to take it easy until the trial was done, so he wouldn't get hurt anymore. But he's continued now. This was from November of 2011. He is still in remission. He's working, playing with his kids, playing tennis, in remission, off drugs. If you look at the clinical response in this paper, and I'm not going to go through all the data, it's all published. What you're seeing in blue is the clinical score, which has a significant improvement from the time the device was turned on until the time it was turned off. This was part of the study design. On day 42, we turned it off, and they promptly got worse. As you're going to see, when you turn it back on, they get better again. But what's important here, mechanistically, is the red line is the whole blood TNF responses in these patients. So we're regulating the nerve signals to regulate the TNF to a clinical benefit. Now, the first patient I showed you had failed methotrexate. That was the first cohort of patients. The second cohort were biological failures. And the New York Times wrote a Sunday magazine article about this patient who identified herself. And she had failed steroids and failed 5 biologicals. She was so sick she couldn't dress herself to go to work some days. And she was implanted in April of 2013. And she is also in remission and now rides her bicycle regularly to the Dutch coast on weekends, nearly 20 miles from her home. And so I am and was over the moon with happiness about the fact that you can actually see some of these ideas help specific patients. And I didn't think it could get any better than that until I got an email one day. It's right after the email from Mary Woolley, if you think research is expensive, and it's right above the email from Diane Quinn that says budget email draft. So this was, this was not a happy day for email, dealing with financing research. But this one jumped out of my screen. Thank you for saving my life. So it turns out that Kelly Owens had contacted me a couple years previously, and I had referred her to the SetPoint clinical trial when she told me she'd had horrible Crohn's disease. Kelly had had Crohn's disease for 15 years. She was disabled by it. She'd lost the ability to be a teacher, which she was a grammar school teacher. And this is the list of drugs that she had tried and failed. And she has the dollar amount of these drugs that she spent over 15 years. She was treated at the Mayo Clinic. She was treated in Hawaii. She was treated at multiple centers in New York. She and her family put together a GoFundMe page and moved to Amsterdam to enroll in the clinical trial done by SetPoint. And that was in July of 2017. She is now 18 months on the nerve stimulator, in remission, taking no drugs. There's a cane in my office that she gave me that she no longer needs, that her father had given her for her 21st birthday. So this is why we do it. This is what it's all about. And for that reason, and no other being necessary, we decided to double down on this concept at the Feinstein Institute and launched several years ago a Center for Bioelectronic Medicine. Now, to repeat what I said before, bioelectronic medicine is a process. It's not a product. It's not a vagus nerve stimulator. It's a new process for developing therapies. You begin with molecular medicine, mechanisms, and mechanistic knowledge of disease, but rather than screen for molecules, you screen or discover or invent neural circuits to control those targets in situ, and then use that information as the specifications to design the technology to target the nerves, to target the device for therapy. I explained this in some detail in March of 2015 Scientific American, and at that time, the list of diseases for which there was already plausible hypotheses and or data was lengthy, and it's grown since then. Ischemia, hypertension, diabetes, cancer, shock, paralysis, and transplantation. This kind of a story has had a tremendous impact. We've raised upwards of $400 million in capital around this idea, sponsored by the NIH, by DARPA. We've set up an important collaboration with the Karolinska Institute some years ago with Jan and Ulf and Peter and others here, and that collaboration has generated hundreds of peer-reviewed publications. We've worked with GE to launch a new program looking at using ultrasound to target neural circuits. We've spun out a company called Sanguistat that's funded in part by Bill Gates' Intellectual Ventures. We last week announced a new company that we spun out from SRI International in Palo Alto. SetPoint I've talked to you about, and we have a very important project with United Therapeutics looking at transplantation biology and pulmonary hypertension. So we are at a very, very important place because we can look hard into the specific details of biologic mechanisms. This idea that is shown here, focusing primarily on immunology, you could replicate this mechanistic slide for cancer, or for diabetes, or for asthma. You can put in your target or disease of indication of choice and map specific circuits back to nerves by developing testable hypotheses and doing experiments that can prove or disprove those hypotheses. It's a tremendous time of progress, but amazingly, this is just starting. This is truly just starting. And so what I'd like to do is just go very quickly through some ideas about science that's happening right now that's moving really, really fast. And I'll go quickly so at the end we can talk in either the Q&A or at coffee if people want to get into more detail about it. So I just made a list of things that I think are incredibly exciting and doable. So first, we're seeing clinical follow-up from 2 years. This is a SetPoint study led by Paul Peter Tak. It was reported off on at EULAR this year and at the ACR. It was selected for a nationwide poster tour. This is the patients who are now 2 years out, showing that they still have a substantial reduction in disease activity and disability that's sustained for 24 months without any untoward safety signals. The data also suggests that biological therapies can be initiated in combination with bioelectronic therapy. There's a whole world of clinical research to do here because patients who had previously failed their biologics, now when they're on the vagus nerve stimulator, they go back on the biologics. Now some of them are responding. So why is that? It's fascinating. The company is called SetPoint for that reason. I think that if we understand these circuits, we'll be able to establish new set points, and that may be evidence of that happening here. There's a fantastic study from here led by Michael Ebertson, who's here today, so he gets to answer the questions on his own study. This was presented at DDW 18 and also at the Bioelectronic Medicine Summit in Salzburg. And this is the Crohn's trials, which are frankly stunning results. Showing here disease activity index, the fecal calprotectin in these patients maps almost exactly to these responses. So what you're seeing are patients who have failed biologics, some of these, again, multiple patients, who are now on no drugs except the stimulator, and many of them are in remission. So there's a lot of excitement about this. These kinds of results have also been repeated completely independently from all of us by Professor Brunet in Grenoble, France, and he also sees significant clinical benefit in Crohn's. There's excitement about doing ulcerative colitis and irritable bowel syndrome for all the obvious reasons. On the mechanism side, we know a lot about how the signals go from the brain to the spleen, but the more we can learn, the more we can advance the field. And one critical unknown question is, how does the brain control, the higher brain, if you will, control the output of the vagus nerve? And we've addressed that using optogenetics. And so my colleague Valentin Pavlov has put a channel rhodopsin, a light-sensitive channel, into neurons of a mouse brain, but only in the neurons that make choline acetyltransferase. Now, anywhere he shines the laser, the only neurons that will fire will be the ones that make acetylcholine. And so here you can see the animals. This is the control for channel rhodopsin. This is the control for chat, and this is the merged image. So you can see that the choline acetyltransferase neurons are also expressing this light-sensitive, laser light-sensitive protein. Now, what's amazing is that he shines the light over the basal forebrain cholinergic system, up behind your nose or the mouse's nose. There's a group of neurons that make acetylcholine, and they project through the whole brain. They're not supposed to project to the vagus nerve. That's what the textbooks say. But when Valentin turned the light on and measured TNF in the blood or the serum or the spleen, just activating a few dozen or a few hundred neurons in the basal forebrain cholinergic system of the mouse blocks the TNF in the spleen. So Valentin has, and he cuts the vagus nerve and the effect is gone. So Valentin has discovered some previously unknown circuit from here to here that controls the immune system. I'm not proposing optogenetics in clinical studies. There's a big controversy about that. That's not what I'm saying. I am saying the more we understand these specific neurons and their mechanistic roles and specific endpoints, it will be possible to make devices to control neurons in a highly specific and selective way, whether it's surgically implanting them or steering current from outside the head. The next question was, how does the signal get from the DMV to the spleen? And that's been controversial. There are friends, and I put friends in quotation marks, who quarterly write articles completely criticizing everything I'm telling you today, so we can talk about that at coffee as well. The big criticism they have is that the textbooks say that the neurons from the vagus nerve don't go to the spleen. And I never said the neurons from the vagus nerve went to the spleen. I said the function goes to the spleen. And so we now have very interesting evidence on how that functional connection occurs. So this is Adam Kressel, who's a MD PhD student in the lab, and he's looking at choline acetyltransferase positive, GFP positive neurons again, but he's taken it to the next level. This is an exposure of the brainstem of a mouse, which is not easy to do. And he's shining the light only on the dorsal motor nucleus of the vagus, the origin of the vagus nerve in the brainstem behind your ear. And then he does a second thing. He puts a cuff on the splenic nerve, which is half the thickness of a human hair in a mouse. So he's got a cuffed bipolar electrode on the splenic nerve, he's got the laser in the brain, and he can record during and after turning the light on in the brain. And this is what you see. So when you shine the light on in the animals that don't have channel rhodopsin, nothing happens in the splenic nerve, because you're looking here at splenic nerve activity. But look at the splenic nerve fires when you turn the light on in the brainstem over the DMV. So there's two discoveries here. The first is that the DMV is the origin of the neurons that control the splenic nerve that a lot of people say doesn't exist. And the second is that those neurons are cholinergic. The signal, that's the first proof that the signal from the brain to the celiac ganglion is actually cholinergic. No, that was not known prior. We're writing this paper now. If any of you get the paper or have gotten it as a reviewer, come see me. I'll buy you a drink. We'll talk about it. It's going to go to a good journal. I'll buy you an expensive drink. This same experiment confirms the function, because when he turns the light on in the brainstem, the TNF goes down in the spleen and the serum. So that's very exciting. We're getting a deepening knowledge of the mechanism of the inflammatory reflex. There's a huge opportunity on the afferent side of this story. And this has been led by Isaac Chu and Cliff Wolf at Harvard in a series of seminal papers they wrote. Remember I told you every cell in the body is surrounded by like a neural net. Most of them are sensory nerves, but sensory nerves can speak in both directions. I said that in the beginning. What they discovered is that sensory nerves can respond to bacterial toxins, and that the signals can actually go up towards the central nervous system, but they can also propagate back down. And when they propagate back down, the signals from the sensory nerve can suppress inflammation in the tissue. And so they've tongue-in-cheek postulated that this may be a way that bacteria have used to target nerves for their own health and survival. In the vagus nerve, I told you it's 80% sensory. We've become incredibly interested, not in this like huge, complex, confusing group of nerves, but which of these nerves might control the signal going up into the brain that Linda Watkins prophesized in her fever paper. And what we've discovered, again, this is being reviewed, it's not published yet, is that the TRPA1 family of neurons, TRPA1 is a channel expressed on unmyelinated neurons that's activated by cold cinnamaldehyde, LPS, and importantly, can also be activated by a drug called Optovin, which is light sensitive. And what Harold Silverman, a recent graduate, doctoral graduate in the lab, showed is that wild-type mice get this nice response in the vagus nerve recording to IL-1, but the TRPA1 mice lose the IL-1 response. To prove the role of TRPA1 in the inflammatory reflex, we took use of this molecule discovered some years ago, 2013, as part of a screen for optically active molecules that would bind to TRPA1. And so this molecule, aptovin, interacts through the cysteine moieties on TRPA1, and when you shine light, the channel opens. So now you can do some cool things. You can take your mouse, you can put the aptovin directly on the vagus nerve and shine the light directly where the aptovin is, and you can generate signals that would go one way or the other, and to find which way the signals go, you cut the nerve and you record up and down. And what Harold has shown is that selectively activating these fibers, which represent probably 3% or less, maybe 5% of the vagus nerve fibers, that's sufficient to stimulate those fibers to activate the inflammatory reflex, because when you turn the light on, the TNF is blocked. If you cut the vagus nerve, you lose the effect. So this is incredibly important because the more you understand about selectively activating selective inputs, you might be able to steer the output in a way that would be optimal to the patient. And here in Sweden, I know you've worried a lot about submarines, and we've been thinking about them too. In fact, we got the idea some years ago, working with DARPA, Sangeeta and I, that it should be possible to treat the cytokine information merely as an electrical signal, and to treat it like a radio wave. Like when you turn on your radio, you hear music, but in between is that music is in a complex form that's transmitted in electromagnetic fields. And so we started doing those experiments, and these have been incredibly important. What we found is that we could do recordings on a mouse vagus nerve, and if the mouse was fasted and the anesthesia was very well controlled, we could get a quiet baseline signal. And this took years to work out the conditions. But having done that, now you can say, what does TNF look like? That's a radio wave. That's a song. That's a TNF song that we've recorded off the vagus nerve. And we played these things to ourselves. We played this back through headphones. And it sounds very different than IL-1. Completely different. So that's obviously an important start, but to move that into clinical development, we collaborated with Theo Zanos and Chad Boughton, and recently published a very well-received paper using naive Bayesian algorithms to isolate and decode the neural activity. And this has been incredible because now we can identify specific groups of neurons firing at the same time in response to a specific cytokine. So that's a huge step closer to identifying a cytokine-specific signal that could be targeted with devices. So using algorithms, it's fun to use algorithms to decode, but if you're a patient, you want more than that. So this patient, his name is Ian, and he was a patient of Chad Boughton's, who's now the director of bioelectronic medicine at the Feinstein. And what Chad did is, working with Ali Rezai and other neurosurgeons, is implant an electrode array into the brain of this quadriplegic young man. And Ian now trained on this device. And Ian thinks about extending his thumb, thinks about flexing his wrist, thinks about extending his wrist, and that's what his brain is doing. That's the patterns that develop from the electrodes in his brain as he thinks about doing these things. Chad and his team then built a computer interface to take these patterns and run them back to specific electrodes to control specific muscle groups in Ian's arm. And now what I'm going to show you in this movie is the first time a quadriplegic human could use his arm by thinking about it. Ian is imagining these events, and his brain is doing it because the brain signals goes back through a computer to control the specific muscles. Chad, presently as we speak, so this is an example of re-encoding the decoded signals. We should be able to do this for the immune system, or diabetes, or asthma. Once we decode them, you can re-encode them, if you know what you're doing. So Chad's presently working on sensors that are connected to an electrode array in the brains of patients in the sensory cortex, so that patients will begin to feel again. Chad wants to restore the ability of a paralyzed person to feel his wife's hand. Here we are at the beginning, and I think we're going to hear this afternoon, I hope, some extraordinary opportunities to go forward for leadership in this field around resolution of inflammation, cardiovascular disease, metabolic syndrome, insulin resistance, obesity, mechanisms for informed mechanisms for sepsis, and potentially wireless, wearable, multi-sensing microdevices, all opportunities for the long-term closed-loop control of chronic inflammation. This is an unbelievable opportunity that is before you today here to talk about. I think that going forward for the next few decades, bioelectronic medicine, beginning with a target and evolving through neural mechanisms to technology that will require new kinds of devices, new kinds of nanotechnology, and new kinds of firmware and AI are going to be the therapies of the future. I cannot count all the emails I get from patients who tell me they want a device, they don't want to take their drugs. And I'm speaking to Michael earlier and others of you who care for patients. Patients don't want to take drugs. They want devices that target evolutionarily ancient mechanisms. This is going to require a lot of time and money. The companies I've been involved with in the last 30 years, listed here, Cytokine Networks, Critical Therapeutics, Setpoint. Sanguistat is a company with Bill Gates to control bleeding, and Neurome is the company with SRI to provide platforms for algorithms and encoding and decoding of signals. These companies have garnered upwards of $1.5 billion of capital, and that's a critically important step to just recognize. However, those that do that have the opportunity to look at the past as the past and look forward around an idea of who is going to do this if you don't do it, and who's going to do it if we don't keep doing it at Feinstein. A lot of places could do this, but it requires a major commitment to what are the current obstacles and how do you solve them, and it requires people collaborating. The one thing that distinguishes the Feinstein Institute that I'm perhaps proud of as much as anything else is that it's a place where collaboration is required, and that becomes really important to success. I think that the opportunities you have to collaborate around your existing resources, to hopefully keep collaborating or collaborate more with the Feinstein are extraordinary, and I look forward to a time when future patients will benefit from all this hard work and fun. Thank you.